All right. Well, hello everyone, and welcome to semester 2 of AP Bio. So, what we're going to be doing today is looking at how organisms control the expression of their genes. When we ended last semester, chapter 17 took us through how to make proteins, and now we know how to, in this chapter, control the production of those materials. So that is the big question, regardless of whether it's prokaryote or eukaryote, is how do we control the expression of those genes? Now, with prokaryotes, we're going to have a situation that's slightly different. Okay, so our focus in this section is going to be prokaryotic control of gene expression. Now, prokaryotes are unique in that they lack a nucleus. So since they don't have any distinct nucleus, the processes of transcription and translation are in essence coupled or continuous. So if you're going to regulate the expression of a gene, regulate whether or not a protein is produced, what makes sense is to control it at the point of transcription. This makes sense visually when we imagine a little DNA molecule here. And along the DNA molecule, you have RNA polymerase enzymes moving and synthesizing strands of messenger RNA. Well, we can see here that immediately ribosomes begin to attach to that messenger RNA and then translate that information into polypeptides or little proteins. So there again, you can imagine if we're going to stop the production of these proteins, we need to prevent transcription in prokaryotic cells. Now, prokaryotes and eukaryotes as well, but organisms can maintain specific levels of end products of metabolism by either having those end products go back and inhibit an enzyme earlier on in the metabolic pathway, which is a quick form of negative feedback, or you can have a slower but more definite control of the production of end products by actually switching off the genes that produce those enzymes in the metabolic pathway, and that's what we're going to look at today. Now, the key player in this process would be the operon. The operon is the system that controls the expression of those genes by switching on or off the machinery needed to produce those proteins. Now, an operon itself is made of a combination of the structural genes that actually produce the protein, promoters, which are the sequences of DNA that are the attachment points for the RNA polymerase that makes the transcript, and the operators, which is a new term here. It's the structure or the sequence of DNA within the promoter to which a special regulatory protein attaches. Now, again, this is only seen in prokaryotes, and there are two major forms, inducible and repressible, and we'll talk about those here shortly. Now, this is a little image of an operon. So we see the DNA in terms of the genes that will be transcribed to make these enzymes used to break down lactose in this case. But you also see upstream of those genes is the promoter, where the RNA polymerase will attach, and then the operator, which is it's good to think of it as like being a switch that can be switched on or off to allow RNA polymerase to attach and then go on and transcribe those genes. And back here is a little regulatory protein that actually produces this key, if you will, that can switch this on or off. So if you're sort of looking at big picture stuff, an operon has three parts: a promoter, an operator, and then these structural genes. Please be familiar with each. All right, now let's talk first about inducible operons. Now, inducible operons are usually seen in metabolic pathways that are switched off. To induce means to force. So these operons are usually switched off, but they can be forced to be turned on when needed. Now, the classic example of this is the lac operon. The lac operon produces a series of enzymes that are used to break down lactose or milk sugar. Now, here we see an image of the lac operon. You can refer back to earlier portions of the Prezi here, where you have the enzymes that are made to break down lactose. We have the promoter, which is where RNA polymerase attaches. And again, RNA polymerase will slide along the DNA and make the messenger RNA. And then back here is this regulatory gene that produces this special protein called a repressor. Now, the repressor, if you look, fits well in the operator. And when the repressor this protein is attached to the operator, we see that RNA polymerase can't attach. So that prevents transcription, so these genes aren't made. Now, this makes sense because these genes are used to break down lactose. But if there's a low level of lactose or a low concentration of lactose in the cytoplasm, would it make sense for bacterial cells to produce lactose? Well, absolutely not. So typically, this operon is switched off. This repressor is attached to the operator, and you don't make the enzymes needed to break down lactose just because you don't have lactose. Now, if lactose is present in the cell, it will have a form or an isomer called allolactose. Now, if we look, this lactose fits very neatly into an allosteric site of the repressor. Now, when that happens, notice how it changes the shape of the repressor. This repressor no longer fits in the operator. When that happens, RNA polymerase can dock to the promoter and then zip right along and transcribe these genes into messenger RNA, so then it can be translated into these enzymes that will break down lactose. So again, normally the lac operon is switched off because there's normally a low level of lactose in the cytoplasm of a bacterial cell. But if you go and drink milk at lunch or at dinner and lactose gets into the cytoplasm of the bacteria in your guts, then that lactose, some of that lactose, will bind with the regulatory protein or the repressor protein, and then that repressor protein gets pulled off the operator, doesn't dock with the operator, then RNA polymerase begins its work, and then you start making enzymes that break down lactose, so you can get the energy from lactose. Now, eventually, when lactose is broken down, you won't have this allolactose bind with the repressor, so the repressor now becomes its active form again, gets linked back in, and you've got the operon switched off. So the whole idea with this is it allows bacterial cells to control the production of enzymes. And if they don't need the enzymes, they're not going to produce them. It helps them save energy and materials. Now, if you think about the type of feedback that is, consider the fact that an end product has control over earlier steps in the pathway. So these enzymes, when they're made, are going to break down lactose, and when that happens, when lactose is broken down, you create an active repressor. So this is a form of negative feedback. Okay? Negative feedback maintains homeostasis. All right, the next type of operon would be a repressible enzyme. Now, to repress means to stop or to prevent. So repressible operons are usually switched on. They're usually making some product, but they can be switched off. And the classic example of this is the TERP operon, TERP meaning tryptophan. Now, tryptophan is an amino acid needed by bacteria. So since they constantly need some level of tryptophan, they're constantly producing it. So the operon is normally switched on, and they're making that product. And that's exactly what we see in this image here. So typically, this repressor is in an inactive form. It won't attach to the operator. So typically, RNA polymerase docks at the promoter and transcribes the enzymes needed to make tryptophan, or the polypeptides needed to make tryptophan. So that's what is typically happening. The bacterial cells are normally producing tryptophan. The operon is switched on. But like we said, it's repressible. If you have a nice big meal, Thanksgiving dinner, and eat lots of turkey, and have higher levels of tryptophan that are in your bloodstream, well, that tryptophan will eventually work its way to the cytosol of the bacterial cell. And when that tryptophan enters the bacterial cell cytosol, you'll see that, it fits really neatly into that allosteric site of the repressor. And when that happens, it changes the shape. Remember, change the shape, change the function. The repressor becomes active, and now it binds to the operator. In effect, that switches off the TERP operon because RNA polymerase can no longer attach to the promoter. So what this does is allow cells to not produce more tryptophan than is needed. So when they need to make tryptophan, they're constantly making tryptophan. But if tryptophan's present, it's going to switch on this protein, and that switches off the operon. Okay, so it's like the key that turns the switch to the off position so that you don't produce more tryptophan than is needed. So let's see here, just other little tidbits. Since tryptophan works with the repressor protein, it's often referred to as a co-repressor. All right, so what kind of feedback is this? Well, again, we think about it. The end product, or tryptophan, helps prevent its own production. So this is another classic example of negative feedback. Finally, we're going to look at upregulation. How can we actually speed up the transcription of certain genes? So we can switch operons on and off using the promoters and repressor proteins. But how can we actually speed up the transcription of certain enzymes? Now, lactose is broken down by bacterial cells as needed. But bacterial cells are going to preferentially break down glucose because they've constantly got the enzymes needed for glycolysis. So when glucose is available, bacterial cells are going to break down glucose first. Now, if you have a situation where you have little glucose but quite a bit of lactose, well then what happens is you've got the lactose there, the repressor protein is switched to its inactive form, and you're going to be making some level of these enzymes to break down lactose. So that switches on the operator. So if you have lactose, it's going to switch the operator on. But if there's little glucose, that actually corresponds to a high level of a molecule called CAMP. Now, don't worry about the names here in particular, but just recognize that this CAMP molecule can dock with another regulatory protein called CAP. And when that happens, it changes the shape of that protein. That protein then can dock on the promoter and help RNA polymerase attach to the promoter and transcribe the enzymes needed to break down lactose. So it's a really fantastic and efficient means of controlling the production of these enzymes. So you have the lactose present, so you switch on the operon so you can make those enzymes. And by using other proteins, you can help RNA polymerase attach to the promoter and make those enzymes more quickly, which is what the bacterial cell wants or needs when glucose is in scarce supply and it needs more enzymes to break down the lactose. Now, if there is lactose present, you're going to switch off that repressor and you can make the lactase enzymes. But if there's not much glucose, or I'm sorry, if there is glucose present, then there's relatively low levels of the CAMP, and if there are low levels of CAMP, then CAP remains relatively inactive. So what that means is RNA polymerase cannot attach to the promoter as easily, and that slows down the production of these enzymes needed to break down lactose. So what that does is it allows the bacterial cell to break down glucose preferentially, and it still allows it to break down lactose to some degree. So again, think of these means of repression as volume control. Your book referred to it as volume control. So the repressor proteins act as an on-off switch with the operator, but the CAMP-CAP system focuses on being more of a volume control. It can speed up or slow down that expression.